We are investigating the role of FOXG1 and DLX5 in the development of a monogamous species under the guidance of our principal investigator, Dr. Sylvester. Our primary research objective is to examine the role of transcription factors in the developing central nervous system of convict cichlids, to understand how gene expression influences behavior. We want to understand pair bonding, display common traits associated with pair bonding such as mutual defensive shelter, biparental care of offspring, selective partner preference, and selective aggression. We are comparing zebrafish, a non-monogamous fish species, to convict cichlids, to know why one species forms long-term social bonds while another does not. We chose two genes that code for transcription factors that induce cell differentiation and migration in the telencephalon. Develops into the limbic system, which contains the prefrontal cortex in the nucleus accumbens. Part of the midbrain develops into the ventral tegmental area, which contains dopaminergic and GABA-ergic neuron projections. Transcription factors mark embryonic structures involved in monogamous behaviors. The mesocorticolimbic dopamine pathway contains both dopaminergic neurons, which are involved in reward and reinforcement, and which can inhibit or excite dopamine signaling. This area is innervated by axons from the ventral tegmental area, which has two-way connection with the prefrontal, and both of those fire on the nucleus accumbens. FOXG1 is the earliest genes expressed in the telencephalon. This and marked in red on the right. DLX5 is a marker of GABA neurons which connect bundles of neurons in the basal ganglia to the prefrontal cortex. In this image, use zebrafish embryo, which shows DLX5 expression in the telencephalon and diencephalon, and you can see characteristics that mark DLX5 in the telencephalon. We want to understand how gene expression influences developmental behaviors. We suspect that interspecies differences in expression of genes FOXG1, a marker of cortical projection neurons, and DLX5, a marker of GABA-ergic neurons, will show differential genes during neural development of convict cichlids. To visualize the expression of these genes, we designed new primers from degenerate nucleic acid sequences from the genome of a closely related species to the convict cichlids. We then used RNA 3' to 5' direction to complementary mRNA sequences in the fixed fish embryos. Antibodies conjugate an enzyme that dye the surrounding domain purple bind to the RNA probes for visualization under a microscope. It's known as in situ hybridization, which was performed in convict cichlid embryos. Join us in the conference room to learn more about the results of our study and to ask questions.